Okay. Okay, sir.
Okay. Very good morning, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, convener of CPGET 2023 free online coaching for all competitive exams in plant sciences, including JL, DL, and Gurukula JL, DL. In this regard, today's our eminent resource person, Dr.O.Padmaja Madam Garu, Assistant Professor, Department of Botany, Tara, Government Degree and PG College Autonomous, Sangareddy. Madam is going to explain about the embryology and the clear and the perfect information and easy going to attempt all the questions regarding of this class. So better you have to utilize this wonderful opportunity because of her huge experience regarding in plant sciences and also embryology. So madam, please welcome and share your screen and start your session, madam. Please welcome.
Thank you, sir, for giving this opportunity. Today I'm going to continue and I'm also concluding this embryology. Today topic. And also we'll discuss some of the bits regarding this embryology. And this is the last topic in embryology. Assistant Professor of Botany, Tara Degree College, Sangareddy. I'm going to continue the remaining part of the embryology. Today our content consisting of structure of seed, disposal of roots and seeds, pollen pistil interaction, and conclusion and the bits regarding the embryology. I think this will be the important topic. We can expect the bits from this topic also, today's class. And before entering that, if we know the theory part of this, the seed. So after fertilization, the ovule will be developing into the seed. And if we see the structure of the seed consisting of a typical mature seed of angiosperm consisting of two parts. We already studied that these gymnosperms and angiosperms are considered as the spermatophyta because they are producing the seeds. But in lower plants like algae and fungi, they are the spore producing plants. But here gymnosperms and the angiosperms, అవృత బీజాలు వివృత బీజాలు మనము వీటిని స్పెర్మటోఫైటా అనేసి అంటాము. దే ఫర్ ద propagation or for the producing the new plants, these plants will give new plants by the seed formation, seed production. And a typical structure of the seed consisting of two parts, the seed coat and the kernel. The seed coat, in the ovule, we already studied that after fertilization, the ovule will be changes to the seed, and the ovule consisting of the envelopes, they may be changes to the seed coat. So seed coat is the protective covering of the seed, develops from the integuments of the ovule. That means the seed coat is formed from the integuments of the ovule, and it is made up of two layers, testa and tegma. These outer integuments and the inner integuments lateral after fertilization. That means after forced fertilization, they're called as testa and tegma. Testa, the outer layer is called the testa, which usually hard and various in appearances, and the following structures are present on the testa. At one end of the testa, there is a scar called the hilum, and the hilum is the position of attachment of seed with its stalk. It is called the funicle, and this hilum is present at the side of the funicle. Near the hilum, a small opening is present called the micropyle. This is called as the opening of the ovule, through which seeds at the micropylar end, they are called as the raphe. And the tegmen is called as the inner layer, which is very thin and papery. The tegmen is much thinner than testa. It may be closely present in grams, remains free in gourds, and sometimes it is absent in the pea. Pea means that are the families of the legumes. And the kernel, the inner part of the seed coat is called as the kernel. It consisting of either of the embryo only, as the ex-albuminous seeds that are present in the leguminous of embryos enclosed within the endosperm, as in the albuminous seeds, paddy and the wheat. The embryo with both endosperm and perisperm, as in cardamom of Gingibraceae. And the latter structure is the embryo. That means the embryo is formed by the fertilization of the male and the female gamete that is present in the embryo sac. And the embryo is the baby plant enclosed in the seed. And it consisting of two parts, cotyledons and tigellum. Cotyledons, these cotyledons are also called as the young leaves, because for developing the small plant, these cotyledons are giving rise for, they're also acting as a first leaves, because they contain the chlorophyll. And their number is either in monocots it is 1, and the dicots it is 2 cotyledons will be present. Sometimes they store food and become fleshy, but in some cases they do not store food and remain thin and the papery, depending upon the storage of the food materials in the cotyledons. And tigellum is the axis. That means it is the main axis, of which one end is pointed and produces out of the cotyledons. That means lie next to the micropylar and is called the radicle. This radicle gives rise to the root system, and that afterwards it will be rudimentary. That means after this part will be giving rise to the radicle, and the other end of the tigellum is the plumule. It gives rise to the shoot system. And in monocotyledons, both radicle and plumule remain covered by the protective sheath. We call that this plumule is covered by the coleoptile and the radicle is covered by the plumule in monocotyledons. And they are the some structures. The portion of axis above the point of attachment of cotyledons is called the epicotyle, that is present above, and the lower surface is called as the hypocotyle. And endosperm is formed by the fusion of this secondary polar nucleus and the one of the male gamete. So in angiosperm, commonly the endosperm is in a triploid in condition, and this endosperm provide food, reserved food of the seed. It may be starchy in wheat, or it may be the oil-like structure in brassica, or it may be in the proteinaceous in the pulses. Depending upon the storage of food materials we are using for the protein purpose, for the cooking of these pulses, or sometimes in the mustards we are stored the oils. We are using as a mustard oil for the massage, or for the small babies for the mustard oils. And in wheat, it giving rise to the starchy like structures, the storage of the endosperm. And the perisperm is the remnant of the nucellus of the ovule. That means the tissue that is present in the ovule is diploid in condition, that is called as the nucellus. And after fertilization, if the nucellus is remnant, if any part of is left, that maybe leads to the formation of the perisperm. And we are also seeing some outgrowths of the seeds. And the seeds of certain plant exhibit special outgrowths to perform special functions. These outgrowths are of different kinds as follows. They are of different outgrowths are seen for the seeds. That may be for the absorption, or that may be for the storage of the food materials, or that may be for the dispersal of the seeds. Like this, the first one is the caruncle. It is the cushion-like structures that are present in the seeds for the absorption of the water. Sometimes the seeds have to absorb water. In order to get the seed coat must be ruptured in order to germinate. This caruncle is a soft structures that are present on the seed for the absorption of the water. And as a result, the seed germination will be taking place. That means that due to this enzyme activity of the germination, So in order to get the enzymes may be activated, water is very, very essential. So in order to store the water, this carbuncle is placing a very important role for absorption of the water in E4BAC, in resinous. ఆమ్దంలో మనం చూస్తుంటాం అలాంటి సీడ్స్ లో అన్నమాట. దిస్ ఈస్ ద అవుట్గ్రోత్ సీన్ ఆన్ ద is known as the caruncle. And the eril, it develops from the chelazal end of the ovule. It is also considered as the third integuments. In meristica, or there may be a ribbon-like structures are seen on the outgrowths. In Pithecanthropum, the eril is spongy and the edible. For example, if you take the commonly we called as the chimachintakaya and santam gada, అందులో seed చుట్టూరుగా మనకు white color spongy structure will be seen. This is considered as the eril. So it is also called as the third integument. And strophioli, it is the outgrowth developed along the hilum of a seed in the dolichos. This is an important bit also. This strophioli is present in which family, they may be asked. That means leguminous. And coma, a tuft of hair present on side of the seed is called the coma. In usually this is helpful for the dispersal of the seeds in calotrophies. In జిల్లేడులో మనం సీడ్స్ లో చూసేదంటే సీడ్స్ పైన మొత్తం కూడా హెయిర్ లాంటి స్ట్రక్చర్స్ వస్తుంది. వాట్ ఈస్ ద మెయిన్ రోల్ ఆఫ్ దిస్ హెయిర్ లైక్ స్ట్రక్చర్స్ as they are acting as a parachutes in order to spread the seeds from one place to another place. They can be easily moving with the air. And wings, it is an outgrowth extended from the seeds as a wing. It helps in the dispersal in the moringa, in oroxylon. And these wings are helpful for the dispersal of the seeds. These outgrowths may be helpful for the absorption of the water, or maybe dispersal of the seeds, or that may be for the edible thing also. Edible means we can eat that structures. And here the dispersals are of different types. We may be seen here. If the dispersal of the wind by the air, it is called anemochory. If it is dispersal by the water, it is called as hydrochory. And if the dispersal is by the animals, we call it as the zoochory. And in zoochory, we are mainly seeing the hooked fruits. Hooked fruits means sometimes the fruits are fixed to our cloths, or to the adheres to the animals. వాటి యొక్క స్కిన్ పైన కూడా అంటి పెట్టుకొని ఉండడం ద్వారా ఏమవుతుంది అంటే these fruits may be spread from one place to another place. And the sticky fruits and seeds sometimes have sticky granular hairs by which they adhere to the grazing animals. ఇవి గడ్డి తినడానికి వెళ్ళినప్పుడు అన్నమాట ఆ స్టిక్కీ నేచర్ గమ్ము ద్వారా అంటుకుపోవడం కూడా జరుగుతూ ఉంటది. ఫర్ ఎగ్జాంపుల్, ఇఫ్ యూ టేక్ ద బర్డ్ హ్యావీ అండ్ ద క్లియోమ్, the cleome itself, the plant itself is, if you touch the plant, it will be sticky. So the sticky nature, the seeds are also, due to the sticky nature, they will fix to the clothes, or they may be fixed to the animals fur also. And they may be fleshy fruits. The fleshy fruits means in mainly fleshy fruits, we usually for the edible means we will be eating that fruits when they are fleshy. So after eating, we will throw the seeds of that fruits. That may be also helpful for the dispersal. And the dispersal by explosive mechanism, that this is a wonderful mechanism we are seeing in the nature. That means here the nature itself helping the fruits for a beautiful explosive mechanism. From the plants. దీనికి మన చిన్న ఎగ్జాంపుల్ మనం చెప్పుకునేది ఉంటే, మనకి గోరెంట పూల యొక్క సీడ్స్ కానీ, ఆర్ అదర్వైజ్ క్రొసాండ్రా కనకాంబరము విత్తనాలు కానీ, ఆర్ రువీలియా ఇస్ ఎ మెడిసినల్ ప్లాంట్. వీటి యొక్క సీడ్స్ కూడా మనం చూసేది ఉంటే ఏంటంటే, వాటిని మనం వాటర్ లో వేయడం తోటే జస్ట్ టప్ శబ్దం తోటి ఏమవుతుందంటే, డ్యూ టు దట్ ఫోర్స్ ద సీడ్స్ విల్ బి డిస్పర్సల్ ఫ్రమ్ వన్ ప్లేస్ టు ద అనదర్ ప్లేస్. Like this type of explosive mechanism we are mainly seeing in some of the plants. So this is called as autochory. autoquery show the following adaptation. In order to have that autoquery type of dispersal in plants, they're having certain mechanism. Touch of some plants cause the ripened fruit to explode suddenly and seeds are thrown to outer great force. If you touch that plants, when the fruits are ripened, if you touch that one, the fruits will be opened and the seeds will be dispersed. Sometimes when they come in contact with water, particularly after the shower of rain, if you observe clearly, when the plants are growing, just like the Crosandra, can you show me? One minute. Sorry. Sometimes when due to this rain water and the water is fall on the seeds, they suddenly burst and the noise like structures, noise like sound will also formed and these are scattered. Example, Ruvelia and the Crosandra. And certain long pods explode with voice, scattering the seeds in all directions, like Bahinia. The Bahinia Valley is also commonly called as the camel's foot climbers. They are very high plants they are. Sometimes due to turgor pressure means whenever this water like substance tissues are present in the germinating seeds, when they absorb water due to this pressure, the seeds will be also burst. Like this, the seeds will be falling from a different distance. As fruit matures, tissues around the seeds are covered into mucilaginous fluid, due to which a higher turgor pressure develops inside the fruit, which leads to the dispersal of the seeds. This is a beautiful, we can clearly see the fruit in the fruit itself, they're having the mucilaginous fluid. The fluid is coming outside and with due to this water and also having this turgor pressure. The Ecballium, Elytrium, squirting cucumber, we can clearly observe this one. And Gyrocarpus and Dipterocarpus, this type of mechanism, like the fruit will be a pressure of fluid is coming outside from the fruit. And next one is the pollen-pistil interaction. In this pollen-pistil interaction, what we are going to study is the pollen grains are coming from the anthers and the pistil is also called as the stigma. And what is the interaction that is taking place between the pollen and the pistil, and what are the barriers are there in order to overcome the self pollination. Like this, we can come under this pollen pistil interaction. So here, that what is the pollen pistil interaction is a sequence of events that occur from the time of pollen deposition over the stigma till the time of entry of pollen tube into the ovule. Just it is the process. The structure that are present towards the left side is called as the ovule. We already know that this ovule is also called as megasporangium, and these are the structures here. And this is the inside this pollen grains. In that pollen grains, we are seeing that vegetative nucleus and the generative nucleus. Later, this spindle-shaped generative nucleus divide to give rise to the male gametes. These male gametes with the 3-celled stage, these pollen grains is falling on the pistil or the stigma. And why this stigma is not helping for the fertilization of this pollen? So what maybe we are seeing that incompatibility, that if the pollen grain is fall on the stigma and the pollen grain will germinate and produce the pollen tube, and this pollen tube contains the male gamete, this male gamete will fuses with the egg. As a result, embryo is forming. That is that, like this we are seeing. But sometimes here the incompatibility, due to this incompatibility, the pollen is not germinating. And if the pollen is producing pollen tube also, if the zygote is formed, the zygote is not the valid in condition, is not having the viability. So this is called as this pollen pistil interaction. In this we are seeing the number of compatibilities. So this is the journey of the pollen till the embryo sac and the egg. So if you see here, the first diagram is showing that, and this is the ovary style and the stigma. So the pollen grains are falling on the stigma. The stigma is also called as the pistil. The total part is called as the pistil, and it is also called as the carpel. These are the terminology we are using in the embryology. And here the pollen grains, after pollination, the pollen grains by the different agents, they are falling on the stigma. And after their adhesion means due to the secretion of the substance from the stigma, they are sticky on this stigma, and they may be due to some water lily-like structures. Due to this hydration, after pollen germination, the pollen tube is formed. These pollen tubes are entering into the stigma, and the entry of pollen tube may be of three types: orogamy, chalazogamy, and mesogamy. Whatever may be the entry, the pollen tube will enter the micropylar, and this pollen tube contains the two male gametes. One male gamete will fuses with the egg, another male gamete will fuses with the secondary polar nucleus. Now, pollen tube grow through the style. Pollen tube enter into the ovule and the embryo sac. In the embryo sac, we are having the egg and the secondary polar nucleus. And one male gamete will fuses with the egg, as a result zygote is formed, and another will fuses with the secondary polar nucleus, as the result endosperm is formed. These are seen in the stepwise in the pollen pistil interaction. And now here, importance of self-incompatibility. So why this stigma is not helping the pollen to germinate, or otherwise if the embryo is formed, that embryo is not viable. Why it is having any importance in the nature? It's helpful in maintaining heterozygosity. Heterozygosity means here different characters in order to attract the other pollens. So when the cross pollination is taking place, will be having the more characters, and that will be having the disease resistant varieties will be also seeing, and the heterozygosity nature helps in evolving the new varieties. In order to get the new varieties, the nature itself is showing that self incompatibility, and which can survive in adverse condition. So incompatible, so new varieties are it can be helpful, and it can be face the different tolerant conditions. That may be the tolerating conditions will also be seen, and prevents excessive self-pollination, which over a period of time leads to loss of vigor. So continuous self-pollination is also will not be helpful. And now if you see this, and what is that why this incompatibility we are seeing is, if you see, there are two types of incompatibility will be there: sporophytic self-incompatibility and gametophytic self-incompatibility. So this gametophytic self-incompatibility is also mentioned as abbreviation as a GS1. And the inability of the gametes from the same parent plant to fuse to form a zygote. That means if the egg and the pollen grain of the same plant also they are not germinating to give rise to the zygote, then it fails to develop. That incompatibility resides on the genotype of the pollen grains. Why they are not forming the embryo? What may be? That means these are with the help of multiple alleles are there. These alleles are not helping the to fusion. So in members like Solanaceae, Papaver, Rosaceae, Rubiaceae, and Liliaceae, in pollen meiocyte, the S gene activated is delayed till the completion of the meiosis. The growth of pollen tube arrests in the style. If you see the diagrams here, the first, second, and third diagram here, in the first one, the two pollen grains are producing the pollen tube, and another one is not showing that pollen tube will be extending, that means rejecting the pollen tube, and that means this is followed by the different types of multiple alleles. These alleles are showing the objection for the developing of the pollen tube in the style and the stigma. And the sporophytic self-incompatibility. So this is due to the secretion of the stigmatic tissue or the transmitting tissue prevent the germination or growth of the incompatible pollen. So the pollen is shed at 3-celled stage, and S gene activation is completed before the meiosis. In the members of Asteraceae and Brassicaceae and Convolvulaceae, the zone of incompatibility is the stigma. That means the pollen either fail to germinate. If the pollen is fall on the stigma, it may be fail to germinate, or the pollen tube fail to penetrate the stigma. Even the pollen tubes are not helpful in producing into the stigma. So this type of incompatibility we are seeing in the pollen pistil interaction. And here the sexual compatibility and the sexual incompatibility. So the sexual compatibility, here we already know the pistil or the carpel or the gynecium has certain devices to recognize the right kind of pollen and allow it to germinate and discard the unwanted pollen. If the self-compatibility is there, the unwanted pollen or the any pollen will be there, that will be recognized and will be discarded, and only the pollen which is suitable for that only will be germination will be taking place. So fertile pollen which bring about fertilization is called the compatible pollen. That means number of pollen grains will be fall on the stigma, and the main one which is fertile and which is helpful for the fertilization will be germinating. So such type of sexual compatibility will be seen. And the sexual incompatibility, here pistil is not able to set seed or fertilize even after pollination with viable and the fertile pollen, then it is considered as the incompatible. That means even the pollen is formed and the pollen tube is formed, but it is not forming the seed. So they may be interspecific or intraspecific type. The self-sterility maybe or the self-incompatibility. If you see the pollen on the here S1 and S2 alleles, if the same alleles are formed, that will be rejecting. If they are different alleles are there, it will be attracting that pollen. So here the pollen grain is having the allele of S3 and S4. So it is attracting the stigma and the pollen germination will be taking place. The same alleles S1 and S2 is falling at the stigma. It is not accepting the pollen to be produced, the pollen grains to produce the pollen tube. So this type of self-incompatibility we are also be seeing here. And the self-incompatibility with the enlarged one, with the diagram which we see, genetically it is under the control of a single gene locus called the sterility locus with multiple alleles. It is maintained by different type of multiple alleles which are also rejecting the pollen grain to produce the pollen tube. So during pollination, the pollen grains and the stigma have the same allele, they remain incompatible. Just the main we have to remember is same allele sunari and reject avutadi anesi. So in the same one, S1 and S2, if the S3 and S4 is there, it will be compatibility will be there and the pollen tube is formed. And in the second diagram, S1 and S2, the same alleles are there. The S1 is not producing the pollen tube. S3 with different alleles, it is producing the pollen tube. And if the S1 and S2, if the same alleles are there, they are both are rejecting. So this type we have to remember that different alleles will be taking place, and if the same alleles will be rejecting. That means the inability of the plant producing functional male and female gametes to set the seed after self-pollination. And if we come to the conclusion and remaining the embryology, we already know the study of embryo structure and development is called as the embryology. And if you see the history of the embryology, the 1682 Grew stated that stamens are male organs of flower. In 1694, Camerarius discovered that stamen are also called as the anthers. They are also called as microsporophylls also in compared to the gymnosperms or the male sex organs, and the ovary is the female sex organs. This ovary is also called as the carpel, and this is also called as megasporophyll. And 1761, Cobrinter found out pollination in flowers by insects. So the if the pollination by insects is called as entomophily type of pollination. And 1824, MEC discovered the pollen tube in Protulica oleracea. That means they first identified the pollen tube. So it may be the bit like the scientist you know MEC. And 1849, Hofmeister found that embryo developed from the cells of the embryo sac. And 1870, Hanstein discovered embryo development in capsella. And the important bit is 1879, Strasburger discovered the polygonum type of embryo sac. And 1898, Navanchin explained the double fertilization and the triple fusion. So this bit we have to remember, the double fertilization and the triple fusion by Navanchin. And 1950, the P. Maheshwari published a book, An Introduction to the Embryology of the Angiosperm. He is also considered as the father of plant embryology, Panchanan Maheshwari. And 1960, Kanta discovered the intra-ovary culture. And the main one is the Guha and Maheshwari produced haploids by the pollen culture. That means the pollen grain culture helpful for the production of the haploid plants. This is an important point we have to remember. And in this also, if you see this, embryology gives complete details about the early development of various plants. We can also know the previous structures of these various plants also. So experimental embryology help us to produce haploids. This is the important point we have to remember. These haploids are produced by the anther cultures, by the production of the pollen grains. And the production of somatic embryos enables the plant breeders to produce artificial seeds. These artificial seeds are very important. These are produced from the somatic embryos. That means that from the somatic cells, the embryo development will be seen. That leads to the production of artificial seeds. And thus embryological data helpful in solving embryo taxonomic also. And it enables us to grow anthers, pollen, ovary, ovule, and embryo segments into whole plants to get the genetic purity. These are also be done under the tissue culture by taking the different part of the flower and helpful for the culturing. And it enables to overcome inavailability of the seeds due to immature embryos with the seeds are shedding. So if the seed is not having the viable embryo, that is not also helpful for the plants to grow. And these are some of the important bits. Main we have to remember is the pollen grains get deposited on the stigma, and they are 3-celled stage. They are deposited on the stigma, and the stigma is having the sugary secretions which are helpful for the stimulate the pollen grains to germination, and helpful for the producing the pollen tube, which come from the germpore by the intine of the pollen grain. The tube nucleus enter the pollen tube first, followed by the generative nucleus. The generative nucleus divides to give rise to the male gametes. That means the pollen tube will having the tube nucleus which are helpful for male gametes direction. It will be helpful for the showing the direction. And after that, one of the male gamete by the triple fusion embryo, the endosperms grows faster than the embryo gets surrounded around the embryo. In some plants, endosperm is completely used as by developing the embryo, As a result, there is no endosperm. If the endosperm is totally used by the developing embryo, there won't be embryo. So in mature seeds, this type of seeds without embryo is called as non-endospermic seeds. They are also called as exalbuminous, and example is groundnut and pea. That this may be an important bit. They may be asking like non-endospermic seeds are also called as exalbuminous seeds. Exalbuminous not having the endosperm. They are mainly seen in the groundnut and pea. And in some plants, endosperm is partially utilized by developing embryo. As a result, there is endosperm in the mature seeds. This type of seeds with endosperm is called as endospermic seeds. They're also called as albuminous seeds. They're mainly seen in the maize and the castor. And in some cases, the epicarp consisting of the epicarp is the fruit. The fruit is having the layers. The fruit layer is called as the epicarp. The epicarp, the outer layer, mesocarp, and the inner layer is called the endocarp. Fruits give protection to the seeds in immature condition and also involved in disposal of the seeds. After fertilization, ovule gets transferred into seed. The integuments of the ovule may be developed into the fruit layers. The outer integuments become hard and forms the testa, whereas the inner integument forms the tegmen. In some cases, nucellus remains as a thin layer. It is called the perisperm. And is the important one. Scutellum is the cotyledon of a grass embryo. It is flattened and is intimate contact with the endosperm, acting as an absorptive organ. This is the main point, absorptive organ. And scutellum plays a key role in absorption of the degraded material during the germination from the endosperm and transferred into growing axis. We already discussed here in monocots embryo, the radicle is covered by protective covering is called the coleorhiza, and the plumule is covered the coleoptile. This coleoptile will give rise to the shoot system, and coleorhiza give rise to the root system. And what are the changes after post-fertilization? This ovary will develop into fruit, ovary wall into the pericarp. The pericarp may be consisting of the epicarp, mesocarp, and the endocarp, and the ovule will develop into the seed. Inner integuments of the ovule will be developed into the outer seed coat and the inner seed coat. Outer seed coat is called as the tegmen and the inner seed coat is called as the testa. And the funicle of the ovule is considered as the stalk of the seed. The egg that is present in the embryo sac will develop into the after fertilization of the male gametes and the female gametes. The embryo is formed, and depending on the embryo, there are two types, monocots and the dicots. Monocot contains one cotyledons and the dicots contains the two cotyledons. And the secondary nucleus. The secondary nucleus after triple fusion give rise to the endosperm, that is also called as PEN. Antipodes and synergids will be disintegrates, and the remnant of the nucellus after fertilization is called the perisperm. The hilum will be remaining as a scar in the legumes. And the formation of fruits without fertilization is called the parthenocarpy. This type will be seen. The production of fruits without fertilization is called as that condition is called as the parthenocarpy. These fruits are called the parthenocarpic fruits. More importance we are having these parthenocarpic fruits in order to avoid the seeds or in order to get the more amount of the pulp. The parthenocarpic fruits are without seeds. They are mainly great commercial value. They are commonly seen in the banana of parthenocarpy. Once upon a time in wild varieties of banana will have bear seeds, fruits. The parthenocarpy could be induced by the application of some of the growth hormones like the gibberellins. And most we are seeing the parthenocarpy fruits in the market like citrus, pineapple, grapes, orchids, and the cherries. Okay. Thank you, sir.
Okay, madam.
Sir,
Right,
thank you, sir.
really, it's a wonderful presentation, madam. Each and every point covered regarding of this CPGET free online coaching, especially embryology part. Madam has clearly explained the total area covered for CPGET syllabus. Really hats up to you, madam. And also the important bits and the history about the embryology, each and every bit, and the discovery, different discoverers and different scientists. We are clearly quoted in this class. So this class is very useful for all our aspirants, CPGET-23 aspirants, and also JLDL and other competitive plant sciences aspirants, also very useful. My dear friends, it is a wonderful class. Really, thank you very much, madam, for your spare your valuable time. And next, we will invite you in another topic. From tomorrow onwards, same time, we will going to meet in this Zoom class, madam. Okay?
Thank you, sir.
Thank you, madam. Thank you very much.